Hello and welcome to the Zoom into Soil meeting. I'm Jack Hannam and I'm President-elect of the British Society of Soil Science, and I'm really delighted to welcome you all to our 6th Zoom into Soil webinar. And today's event has been organised and supported by one of our regional groups. This is the Northern Soil Network, and that's chaired by Dr Manoj Menon. And he will be leading the question and answer session for the webinar today. So before I welcome our two speakers, it's going to be a really exciting session. I just want to briefly introduce you to the British Society of Soil Science as hosts of this webinar. We're an established international membership organisation and charity, and we're committed to the study of soils in all of its widest aspects. So we have members working in academia, but also within industry, so practitioners of soil science, and also for other members who have just a keen interest in soils. And this year we'll be running 10 Zoom into Soil webinars, and the next one in the series is actually going to be hosted by one of our publications, one of our journals, the European Journal of Soil Science. And the next one is going to be on Wednesday, the 9th of June. And that's going to be showcasing the work of early career researchers. So that's going to be really exciting to see what new research is coming out from these members of our organisation. So do look at the website for further information about that event on the 9th of June. So as you can see in the slide here, just some basic housekeeping before we start, before I introduce our speakers. There's lots of you here today. It's really exciting to see you all joining. You've been muted, and we'll be taking questions at the end of both presentations. So there'll be 2 presentations, and we'll be asking questions on your behalf at the end of both of those. So please, could you submit your questions? You can use the function in the webinar to submit these. And please do this before 12:50 to allow us to get through as many questions as we can. And Manoj will be asking them on your behalf after the presentations are finished. So although there's a raise your hand button, please don't use that unless your presenter specifically asks you to do that. So you can ask the questions obviously at the end in the chat. And just finally, for those of you that are members of specific organisations, the presentations have been awarded CPD points for both BASIS and the National Register of Sprayer Operators. So just contact us after the event to be able to claim those CPD points. And as you probably are aware, we're recording this session, so if you want to watch it again, you'll be able to see it on our YouTube channel, and there's also all of our other Zoom into Soil seminars as well that have also been recorded, and those are also on our YouTube channel too. Okay, so that's enough of the introductions. I'm really excited about this webinar today. It really literally is zooming into soil, so looking at those sort of hidden processes and structures in those soils that we're not necessarily aware of. So I'd like to introduce you to our first presenter, and that is Dr Carsten Müller. And he is an Associate Professor of Soil Science in the Department of Geoscience and Natural Resource Management at the University of Copenhagen. So this is really exciting that he can join us from Copenhagen today. And his main research focus is understanding the fundamental biogeochemical connections between plants, microorganisms and soils in both managed and natural systems, and from poor, so really tiny, to field scale. And he combines physical, biological and chemical analyses to gain a deeper understanding of the sort of energy and fluxes in this really complex 3-dimensional soil system. Majority of his research areas is actually in the cycling of organic matter and in soil structure that has been formed by interactions with plants. And he's expanded these kind of techniques by using sophisticated imagery and image analysis with a focus on nanoscale secondary ion mass spectroscopy, so NanoSIMS for short. And this can then study at that very small scale these intact systems and the relevant processes happening at those scales. So he's looking at how via roots, plants can actually regulate all of these sort of important processes like carbon sequestration and nutrient cycling in soils. So the results of this work, a really important prerequisite for developing future sustainable agricultural management practices, to preserve soil health, but also look at increasing carbon storage in soils. So I'm really excited to see Carsten's presentation, and over to you. Thanks very much.
Yeah, thank you very much. Thank you very much for this very kind introduction, and that's yeah, it's a pleasure to get the opportunity to be part of this nice seminar series. So thanks for the invitation as well. Yeah, I will basically take you to the microscale today. So, and I took that really like in a practical note, like zooming in, and I want to take you to where I think most processes, or where actually most processes in soil happen, and this is really the microscale in the soil. And soil processes are regulated at the microscale using microscopy. Microscopy, it's only part of what I will show, but it's, I guess, the catchy way to start with it. And where we start is just having a look at the little clumps of soil. And this is a soil aggregate. This is what we call a micro aggregate. It's larger than 250 micron, and this stems from a rhizosphere. So that stems from the zone in the soil that is directly affected by the organic matter, by the carbon, nitrogen that enters the soil from the root. So it is a place in the soil where you get a lot of carbon in, and where you have a lot of microorganisms sitting, because there's this fresh carbon coming in. And that's also the reason why this is a zone in the soil that's highly active, and it's of higher relevance for the function of our soils. And if you look at this lumps of soil, and this is, go out in the garden and you find that and make your hands dirty, and this is what you have on your hands. But if you zoom in, if you do exactly what this series or this day is about, if you zoom in, you see that's like Serengeti. There's like a vast number of microorganisms. This is particularly true for this kind of samples from this hotspot. So this comes from the rhizosphere. This comes from a place where there's a lot of organic matter, there's a lot of, so to say, food, and then you end up with this zoo of different kinds of microorganisms. And this is where a lot of biogeochemical functioning of those soils is regulated. My background is most in soil organic matter cycling and the persistence of soil organic matter, fractionation, what stays in the soil for how long, and how is this composed, what's the chemical composition. But all of that is regulated at this scale, at the microscale. And there was this paper last year from Johannes Lehmann and co-authors, and they were looking in the functional complexity of soil organic matter cycling. And one, besides the temporal and the chemical composition, or the temporal dimension and the chemical composition, it is the spatial heterogeneity in the soil. That means there's a way stretch where your soil lumps don't look like this. It's like a desert. But then there are places where there is a lot of food and there is a lot of microorganisms. And so this spatial heterogeneity, this affects carbon nutrient flow, but also how much the microbial community gets back when the microbes invest something. And this all regulates the persistence of carbon in soil. So how much carbon is there, how fast does it turn over, but with that also plant nutrition. So how fast do plants get nutrients back from the soil organic matter? And this is all scale dependent. So size matters for organic matter cycling. And if you start on the left-hand side with those two larger bubbles, we have conceptually and also practically two major components in our soil. So we have the major input, that's plant residues, that's plant-derived organic matter, and this is rather on the larger scale. And then as soon as you go and then related to those aggregates on the right hand side, those soil lumps that I showed you before, they can be really, really tiny. And the more tiny they get, the more you have something that is microbial derived, microbial residues. Primary, that's plant material, but then it gets transformed by microorganisms. And so it's all scale dependent. It's all dependent on where does this happen in the soil. And I want to take you on this little journey, at least showing you two major parts of that, how that gets into the soil and where microbes are active together with the plant input. That's Rhizosphere, and this is the Decomposition. So actually that plant material that gets decomposed or turned into soil organic matter. And this also leads to the two major pools, the two major fractions of organic matter in soil. So there is particulate organic matter in the strict sense, a particle. So what you see here, this is a scanning electron micrograph. So we are still in the microscopy. So this is all like microscopic techniques that I kind of like weave in this illustration of what we are talking about. What you see is, you see plant structures. This is really resembling like plant cells and so on. But on that, you also see fungal hyphae. These are particulate organic matter. And then you have this other great pool you might have heard of. That's mineral associated organic matter. So you have mineral surfaces in your soil, be it especially clay minerals, be it iron oxides, and on those surfaces you have carbon or organic matter sorbed, and those two are the major pools. Plus, you of course have dissolved organic matter in between, and also as a means of transport. So this is what we're looking at, and again, we are at the microscale. This is, of course, it has implications at larger scale. So if you speak about CO2 emissions, if methane, N2O, whatever, but also nutrient cycling, that all goes down to those little entities in the soil. And of course, depending on the soil type, depending on the region where you're in, depending on the climate, this differs. So your soils are defined by kind of like the balance between the two, and then again regulated in a spatially manner due to soil structure. Microscopy, zooming into soil, and I mean classical microscopy, and I started with a scanning electron microscope, but just like your bare eye, or you all might be familiar with normal light microscopy, but there's much more now. There's means of mapping elements, there's means of mapping isotopes. So there's just structural microscopy in terms of what I showed you before with the scanning electron microscopy. And this graph just to highlight, there is again a scale into it. So you go from large scale techniques where you can analyze larger specimen, and we will later on get a talk on the micro-CT. There is a specific spatial scale that you tackle, but then also you have the ability and the possibility to get more information, not just the structure, not just like I did before. I showed you microbes sitting on the soil aggregate, and you just see like the structure, but you can also look into how are they composed, what's the actual composition of them, and I show you something later, especially with respect to isotopes. Classically, that's also like on top of classical, but it's like this is light microscopy, and what you're looking at, this is like the string-like thing. This is a fungal hyphae. So it's a fungi growing through the soil. You see the soil minerals around it, those spheres around it, and on top of this fungi, there's little bright dots. Those are bacteria. So what you basically look at is a fungal hyphae. So you have this fungi growing through the soil, and with kind of simple microscopy and staining, so introducing chemical that stains it, that makes it fluorescent in fluorescence microscopy, you can find those spots. And then you might think of higher resolution work, and going back to Foster in the early days, and those great images, and that might be a bit noisy when you first look at that, but what you basically look at, there's a really thin section of soil, and what you see here, like in the down in the part where it's indicated with B, these are bacterial cells. So and where you have the C, this is clay minerals. So you have this bacteria sitting directly at the clay minerals, and this is really just like the imaging part of it. So where I said, like, you get the structures, you get the relation of this biological entities and the minerals, and you could assume, or you could assume processes from it. But where it gets really into it is when you also try to get elemental composition, isotopic composition that might give you more answers to actual processes. So, and one way to do that, and here I now, and Jack was already putting that on, nanoscale secondary ion mass spectrometry. This is one technique that's, I think, around like 20 years now. Started off in cosmic chemistry, and then material science and so on. Geology, it's big, but this is an instrument that gives you the opportunity to image isotopes, and especially for soil science, it's great because it has a high spatial resolution, and it can give you the distribution of 13C and 15N. So this is an example. The gray parts that you see, these are the microaggregates. These are the small mineral stuff in your soil, and they contain carbon. This is the mineral-associated carbon I was referring to earlier. And you see there are hotspots. So it's not like the color indicates on the left that's 13C, on the right it's 15, and you see it's not evenly distributed. It's patchy. So again, there's this spatial heterogeneity even at this really micro scale. So on top of having structural information, having the information that's a microaggregate, you can now go on and say, okay, where does and this is Lucerne plant material, where does this end up in this particular soil fraction? But this is not just images, there's data behind it. So I show a lot of colorful images today, but of course you can do image analysis from it. And what I circled in, these are the outliers, and this is like where you get the fresh organic matter sitting in this fraction. So actually the outliers here are good because they indicate where your fresh plant material ends up in this fraction. And then I said, we go at the interface, and this is what you look at, again, a false color image, scanning electron microscopy image. This is a root surface. Those spines sticking out, these are root hairs. In reddish, that's presumably microbial residues, and bluish, these are soil minerals. This is the interface where the root meets the soil, and where I said before, this is where a lot of microbial activity happens, and also the organic matter turnover in the soil is really determined to a large extent. What we see if we look into like actual structure formation, or at least aggregate formation, or what we can retrieve as an aggregate from the soil, we see that roots have this positive effect on structure in the soil. And the idea is, and there we have good indications for that from other analyses looking into specific microbial communities, we see, especially for subsoils, that roots foster this aggregation process. And so that's also the question, does this on the long perspective foster carbon storage also in the subsoils? But this just as an entry port saying, okay, we know there is this positive effect on aggregation, and we also know from, again, the PLFA analysis that fungi play a large role in it. And now again, using this nanosims, and what you're looking at now is, and you see this in the scaling electron microscope graph on the left, this string, this is a fungal hyphae again, and detached to it, they're small microaggregates. And then and you see this image where you see like green, the microbe or the organic matter, and then blue and red the minerals. And then we also use the tracer here, 13C, and then you see this little spot on top of this hyphae. And this is again bacteria. So this is a different view on this fungal hyphae. I showed it in the beginning with this fluorescence imaging, and where you say, there's bacteria on this hyphae. But now you can say, those bacteria on the hyphae, they consume more. There's a difference in the consumption of root-derived organic matter between the fungal hyphae and the bacteria on that hyphae, and then together with the structure build up, seeing those aggregates attached to this hyphae. So it all comes together in those hotspots, and it kind of like all comes together also who's doing what, fungi versus bacteria, and then ending up in regulating this biogeochemistry or aggregation in this sense. And then just as earthworms and soil fauna gets more and more, and for good reasons, more and more attention. And this is work by Alex Vidal from her PhD thesis. What she did is she used earthworms, or she looked into how do earthworms turn over or decompose shoot and root material differently, and she looked all sorts of things, but she also looked into the casts, the earthworm casts that were produced using TEM. So this, what I showed you, that the image from Foster, these great images where you see, like here, the plant cells, the minerals, and all together. And she combined that in a nice way with nanosims. So the plant material was labeled with 13C. So there's this opportunity to follow the plant material into those casts. You get the structure information from the TEM, from the transmission electron microscope, at a really high resolution. And that gives you an idea, how is this material transformed? How are differences between the shoot and the root material? And so at least for the early stages, it was clear that the chemical composition of the material, so cast versus, or shoot versus root, really drives this organic matter formation in those casts. And that was my my link to go to the Decomposition, like away from the root, but also to have a little bit soil fauna in. And with that, going really to the Decomposition. And this is a piece by Christina Witzgal, PhD student of mine, and where we looked into the decomposition of plant material in differently textured soil. And the main idea was, okay, how does this work? We bring in this particulate organic matter, this plant residue, and how is this decomposed, and how does this end up in presumably more persistent carbon pools, like mineral-associated carbon pools, or carbon that is entrapped within aggregates. And as I showed you before, it's all like at a really small scale. We use two different types of soil, or we use the loam and we added sand to make it more sandy. So we try to have the same microorganisms in that game, and then we had this depth gradient in it, and really like sample those little layers to get kind of like a resemblance of how soil development could work at this small scale. And what became clear is, again, that fungi played a tremendous role in the decomposition and in the turnover of the material. And this is data that you look at based on PLFA, so the phospholipid fatty acids, so the outer part of the microorganisms that's specific for certain microbial communities. And from that, it was clear most of that plant-derived organic matter was ending up in fungi. And then the question was, okay, how does this relate to the structure? And this is one of the larger aggregates, a real like a macro aggregate from this experiment. And what you look at, like the bluish stuff, this is really like mineral dominated, and the brownish, this is particulate organic matter. This is a piece of this labeled maize litter. And then if you look closer to it, this is what happens on the maize litter. So in the base, in the background, you see this decomposing plant litter. And on top is, again, it's partly a zoo. You see fungal hyphae, you see singular microorganisms. There's a lot of fungal spores, but also a lot of minerals. So directly at this biogeochemical interface, the interface between the plant material, like in the root, like in the rhizosphere, and the mineral soil, this is where everything happens. And we had a particular look, again, with the nanosims at this surface, and not embedded, just plain, just look at it, like I zoomed in before on this, like at the early beginning of my talk, zoomed in on the surface, and we did this with the nanosims. And then we ended with those really colorful images, and being able to resemble the plant residue, the composition, like 13C plant residue composition, the fungal hyphae, the bacteria, but also the extrapolymeric substances, or the, like you could say, biofilm in between, that kind of like cloaks the whole system. And via image analysis, you end up with like real numbers, and you can say, okay, how enriched is this material? And you go here from this particulate organic matter to like the fungal hyphae, and you can nicely show there are clear differences in the consumption and turnover, and in the role that the single players have in the decomposition of the plant residue. And you also see, and this is something that's nice for working at the microscale. If you have a classical bulk sample, you grind that up and you get one highly accurate and also highly important value, but you just get one value. Whereas if you do microscale imaging, whatever that might be, like bare eye or microscope or something like this, like nanosims or so, you get the variation. This is like you zoom in on a plot outside on the field. So you get more variation and more like a resemblance of the heterogeneity. What you also, so this is this, and the other thing is you clearly also see, and this is like the elemental imaging component of that, you see this shift in the CN ratio, and I forgot to mention that earlier. You start with wide CN ratios in the plant material, and then the more you go to this microbial dominated material, the CN ratio gets narrower and narrower. That's just due to the like constraints in the buildup of the organisms, from plants to microorganisms. So, and this is possible with this approach. And what we basically did here was showing that this particulate organic matter, this is not just a substrate. This is not just something that you refer, that's label material. That's like, yeah, that's gets eaten up, and let's focus on the mineral associated organic matter because this is stable. No, it's a functional entity that is tremendously important for both the buildup of aggregates. This is relatively, like, that dates back to Oates and so on, and Claire Chenu worked on that and so on. But then also directly on the formation of mineral associated organic matter. So this is also a hotspot for like building up persistence of organic matter in soil that you can image at this high resolution. And with that, I hope I kind of like brought you into it, and I kind of like attracted you to images. And I want to finish with this fungal hyphae. This is a fungal hyphae. Basically, the strings, the brownish stuff, this is a fungi, and all the colored organisms there, that's bacteria on that fungi, and having fun and sliding around. And the thing is, using this high resolution imaging and spectral microscopic analysis, and nanosims is just one part of it. There's a load of different techniques. This can support results, and I want to really stress that point. I think it is meant to support classical bulk analysis and concepts. I wouldn't. That's not made for like replacing it or whatever. You need quantitative data, like what we are used to in soil science, like gram per kilogram or tons per hectare, something like that, you can work with, and that's important for people out there in like practical soil science or like farmers or whatsoever. But the underpinning for the mechanisms and the processes, for that you can get nice support from this high resolution imaging and microscopy. Want to thank a lot of people, so some of them in the pictures, but a lot of colleagues I worked with over the years. Of course, also a lot of funding involved in that, and with some pictures from Western Denmark, where I'm actually at the moment not in Copenhagen. I'm in Jylland, so Western Denmark. And with those pictures, I want to say thank you.
Thanks very much, Carsten. Absolutely fascinating. Nice podzol in the picture there as well. Hope you've done some soil profiling while you've been there. The amazing viewpoint of these microscopic processes, but not just imaging what actually is there, but understanding what are those bacteria and fungi actually doing? What are they? How are they involved in carbon cycling? It's an amazing technique to further our understanding of some of those processes. So yeah, thanks very much, and we'll be taking questions. So if you have, I'm sure there'll be lots of questions. Please use the question function to ask your questions to Carsten, and we'll be asking that. Manoj will be asking those on your behalf at the end of the second speaker's presentation. So yeah, thanks very much, and we'll see you at the end of Tinashe's presentation. Brilliant, thanks a lot. So I'd like to welcome our second speaker. And that will be Dr Tinashe Mawodza. And he's going to be starting a fellowship at Nottingham University, working on a project called Designing Future Wheat. He's going to be investigating in that project the genetic and environmental factors to determine the ability of wheat roots to locate soil pores and elongate to depth, obviously to find nutrients and water. His main research is though centered on exploring ways of improving global agricultural productivity. So this project is obviously very relevant to that, but without compromising environmental quality. Tinashe previously worked as a research associate at the University of Sheffield, so he was part of a team investigating how to improve soil salinity and sodicity management using sustainable irrigation practices, and this was in a study area in Turkey. And prior to that, he completed his PhD also at the University of Sheffield, where he was looking at plant-soil interactions, a bit like some of the work that Carsten was looking at here. So investigating the impact of plant water use on the root architecture. So you saw some of those images that Carsten showed, and also the structure of the soil as well. He has a master's degree and bachelor degree in soil science from the University of Ghent and University of Zimbabwe. So the focus of his talk today will be primarily on the work that he conducted at Sheffield as a postdoc and a PhD student. So I'm really delighted to welcome you, Tinashe. Over to you, if you'd like to share your presentation. Great. Thanks very much.
Can you see my presentation now?
Yes, I can. That's great. Thanks.
Perfect. Hi, everyone. Thank you, Jack. Thank you, everyone, for taking your time to be in attendance. So I'm going to take you through parts of my research. It's kind of like a continuation from where Kirsten left off, in the sense that instead he was looking at the more nano scale, but I'm zooming out a little bit and looking at the roots and the soil and how they interact. So I'm just going to demystify the hidden half, which is mainly the roots that are growing underground, and how the interactions with the roots, and how that plays a part in determining our yields in most of our soils. So in my talk will be divided into 4 different sections. I'll begin with introducing the motivation behind my studies, and then I will explain the principles and the techniques behind some of the discoveries that we have discovered. And then finally, I will take you through some of the research results that we have got through some of these techniques, and then I will shed light on some of the advantages and disadvantages of some of the methods that we use, and some future experiments, and how they may be penned out. So to begin with, I'll start with a poll question. The first question that I'll ask you today, this is kind of builds up to what I want to talk about. How tall do you think the tallest tree is? How tall do you think the tallest tree in the world is? Please select your answer.
Tinashe, I don't know whether you can see that, but the poll has come up as 67% think it's 115.85 meters.
Okay, perfect. Yeah, so I see most of you actually know the answer to this question, which is actually quite great. So the coastal redwood, which is often named Hyperion, which is about 700 to 800 years old, is a staggering 115 meters from the surface of the soil. To put that into perspective, it's actually much taller than the Statue of Liberty, as well as taller as well than the Big Ben in London as well. Okay, to move on, I have another second question, which I would want you to answer again. So how deep do you think the deepest measured roots of plants are? How deep do you think the deepest measured plant roots are? Would you please select your answers?
Okay, so on that one, pretty close, but 38% in the end say 68 meters.
Okay. Oh yes, I'm glad some of you got that right. A relatively small shepherd's tree that was found in Botswana, it's similar to the plant shown on the slide there, which is in the Kalahari Desert, was the deepest ever measured plant root, which was at a depth of 68 meters. To put that into context, this is similar to the length of a Boeing 747, which is the jumbo jet. These roots were actually found by accident during a well digging expedition in the deep sandy soils of the Kalahari Desert when they were looking for water within the soil surface, beneath the soil surface, sorry. So this kind of puts into context the ethos of our research. So most of us can actually appreciate the top part of the plant. However, in truth, it is nearly impossible to tell how deep the deepest roots are because of the opaque nature of soil, which is not transparent. This is similar to looking at icebergs from a boat or a ship. When we get the expression, the tip of an iceberg is only the top half of the iceberg that is not submerged in water is seen. And then there's usually a massive area of iceberg that's actually underwater, which we actually can't see from the top. Let's say if we are on a boat. However, if we would submerge ourselves, we would see quite a big chunk of the iceberg within the water. So similarly with plant roots as well, on the other hand, one of the major drawbacks that we have is, unlike with icebergs, where we can actually get into the water to actually visualize the iceberg, with plants in general, we can basically only see the top parts, like the canopy and the tree leaves and tree trunk, which we normally associate as being just the plant, but there's a whole world that's just around, just down from the ground from where we would be, and it may be as extensive as 60 meters, which is a full jumbo jet that goes down right deep into the soil. So our research, basically, when I was a PhD student as well as during my postdoc time, is trying to entangle these interactions between the root and the soil that are occurring just below the surface. So this has the consequence of actually improving our understanding of plants and actually enabling us to show how these interactions help in determining yield of the plants that we grow. So I think in my title, you actually had me talking about the hidden half. So roots are often referred to as the hidden half of plant growth, and I think we have like two or three books that talk about this phenomenon. So because they often occur in the opaque soil, they're actually unseen, and 9 times out of 10, we actually don't appreciate how diverse and how rich the diversity of roots within the soil are, although these are an integral part of plant growth and our yields within our different environments. Moving on, so there are different methods that have been used to show how plant roots and soil interact. The most rudimentary methods involve digging up the soil, like for instance in the picture on the extreme left, where people actually dig up the plant and actually shake off the roots or wash off the roots, and then study the roots from there. There are also other more modern techniques such as aeroponics, where plants are actually grown suspended within the air, and they are given a mist of water, which is gaseous, so we can actually observe the roots. And then there are other more modern actually tools, such as studying it in axenic culture or in agar, where plants are actually grown on the surface of a hydrous media, such as agar in our case. And then in recent times as well, there are also people who've been growing, especially vegetables in horticulture, plants in water laden with nutrients. And then the most recent of these has been the growth of plants in what they call transparent soil. However, this is really not soil because the transparent soil that they call transparent is actually a polymer called naphthalene, which has similar properties to soil, but isn't really soil, but has the unique properties of being able to be a bit transparent, and such that we can actually visualize how the root is growing within the soil and appreciate the root architecture of the soil, which is essential for yield. So in my research, in the research in our lab, we use non-invasive imaging that may be common to most of you, and most of you will definitely have seen in action at the airports, where we have X-ray scanners that actually scan people and scan people's bags for different artifacts, looking for any contraband that may be in those bags. However, there are other methods that I shan't dwell on, such as magnetic resonance imaging and positron electron tomography. These I won't dwell on because we normally focus on X-rays and neutrons for studying of soil. So in terms of X-ray and neutron imaging, these two are kind of complementary to each other. With the X-rays interact with electrons, as you can see on the image on the extreme left. So they are sensitive to elements that are quite heavy or dense in terms of neutron density, such as those lower down the periodic table. And then on the other hand, neutrons kind of interact with the nuclei, so they don't necessarily are not necessarily sensitive to elements that are lower down, but have like a random distribution of elements that they're actually sensitive to, and they're also sensitive to isotopes of different elements as well. So neutrons are mainly suitable for studying of roots, as well as water, because they're actually more sensitive to water and organic matter, whilst X-rays are more suitable for study of soil structure and the relationship between roots and soil structure in general. So in terms of the basic principles of how these methodologies or methods are governed, these are governed by what is termed the Beer-Lambert law, which is actually funny because it was developed by Pierre Bourget instead of Lambert and Beer, but Lambert and Beer have the name that is normally used for these techniques. So basically, what we do is we pass through the X-ray or neutrons through the sample, and when the sample interacts with these X-rays or neutrons, some of the beams or the rays are actually transmitted to the detector. But what we normally are interested in is what is absorbed by the sample, and then an element of noise actually comes from the scattering of some of these rays when they actually interact. So the difference between what is transmitted initially and what is actually recorded on the detector actually gives us the image of the X-rays that we have seen, I'm sure most of us. To begin with, this was a good measurement initially, but later on within the evolution of X-ray imaging, they actually wanted to see actually within a particular object or item in 3D instead of just in 2D, because yes, in 2D we can get an appreciation, but it's difficult to actually tell which is which and what is where. So these two gentlemen over there who actually won the Nobel Prize for their work with X-ray CT imaging actually developed techniques and skills that are actually able to combine all of these split-second images from the X-rays, and then they rotate the sample or whatever plant you want to image over 360 degrees, and then or 180 degrees, depending on what you actually want. And this will be then reconstructed into a 3D image, which we could actually see on the scan on the right of the screen. So the rotating of the plant while taking images was the most important key element that they actually introduced, which actually enabled us to visualize plants in 3D. So in terms of my research so far, I focused on 3 different plant species. For instance, the Arabidopsis, which is the model plant for genetics in plant science, as well as wheat, which is basically a bread wheat, and then rice, which is a common plant in most tropical areas. So in my research, I mainly focused on water use efficiency, that was derived from reducing the stomatal density of plants that we imaged. So to begin with, I'll just look at wheat, which is like one of the most popular plants within the UK. So in terms of wheat, we actually used some mutants which had a reduction, mutants in the gene which actually reduces stomatal density, as I mentioned before. So we could clearly see and observe what was going on top of the surface, but we couldn't actually ever get an appreciation of what was going on below the surface. And then the consequences of these mutations were actually almost unknown to us. So my research, my PhD, was actually to actually disentangle the relationship between the soil and the plant, and then trying to understand how the plant roots differentiated themselves within the soil. So I used 3 different genotypes. So the wild type was Fielder, and then the 2 other genotypes had mutations in the EPF1 gene, which controls stomatal density, and that's because I overexpressed it. It actually reduced significantly the stomatal density of these plants. So in terms of my results, I actually got the following images. So this is just a snapshot of some of the results I got. This is just an appreciation. So as you can see, there are like 3D images of the plants I had. So in terms of what I actually got, the significant reduction in root length and root volume and root biomass and water use efficiency was actually discovered at 5% level significance. However, there was no significant difference between the 2 individual stomatal mutants. We could actually kind of conclude that there was a difference. So the mutation of the stomata, reducing the stomata, actually played a part in what was going on below ground, and this may actually have consequences in terms of people trying to actually grow some of these mutants in the field, as maybe they may not have a strong enough root system as maybe we would want. In my next experiment that I'll talk about, I will also focus on wheat as well. So with this one, instead of using X-rays as the previous example, I actually used neutrons. So this neutron imaging, I actually was able to map the different root architectures. So because the neutron imaging is normally done in very, very small containers, what I ended up doing was actually growing the plants in smaller, very, very small containers. Out of something that was not by design initially for this particular experiment, we actually noticed that the aggregate segregated, the large aggregates segregated to one side of the tube that we were growing, and then the other side, all the finer elements. And we could actually notice that there was a distinct difference in terms of the growth patterns of some of these roots, such that when the roots were actually growing in the coarser aggregates, they actually were longer, but there are actually fewer roots that were growing there. And then on the other side, when the aggregates were finer, there were more roots, but they were a bit on the smaller side. So to investigate this further, we actually had another experiment, which we actually split the aggregates into distinct sizes, with 2 to 4 millimeter aggregates on one side and 0.25 to 0.5 millimeter aggregates on the other side. And then we grew the plants within the middle, trying to discover which of the 2 different sides of roots that the plant would actually prefer. And to confirm the findings from our previous experiment, we actually found out that the plant roots actually preferred the finer finer aggregates, and actually grew longer but almost straight without any lateral roots within the coarser aggregates on the other side. And then some of my other research focuses on, as I've mentioned before, Arabidopsis, which is a model plant. So with Arabidopsis, which is a relatively small plant, it's a very small root, which I actually tried washing this particular root and trying to determine how the roots would actually elongate within different samples. But this was kind of impossible because the root is quite friable. It's very small and friable. I think the roots are less than 100 micron, which is like almost like the size of your hair, which makes them much difficult to work with, and washing them almost becomes impossible. So in my research, I actually focused on imaging these. So using the CT, X-ray CT and neutron CT imaging, I actually was able to pen out some of the roots and how they actually expanded throughout the soil, and actually mapping the 3D architecture of the roots, and trying to discover how some of the water use efficient mutants that are used and how they actually interact with the soil. Yeah, some of the results are actually still not out yet. I still am preparing some of my my work to publish this, but these are included in my thesis as well. And then we also investigated the erenchyma of rice roots, where we were able to investigate how the erenchyma actually responds to moisture variability under different water regimes. Again, this was kind of a unique research area because we actually looked at the full length. So most people who actually research and the erenchyma or the air gap within the roots of rice actually mainly focus on specific areas or very thin slices. But in our research, because we were using it in 3D, we're actually able to discover how the changes in erenchyma throughout the root system was going to affect plant growth, and how it responded to the different watering regimes that we had actually executed. So to conclude, I would like to highlight that non-invasive imaging, the tools that are used, which are the X-rays and the neutrons, are actually a powerful tool to reveal some of the hidden interactions that are actually occurring below the surface of the soil. And there are numerous advantages to these. Firstly, the neutron and X-rays, for instance, ionizing radiation, both of them are ionizing radiation, which may actually disturb plants and actually limit us continuously monitoring plant growth. And then they are also having trouble in terms of resolution as well, because they can only visualize a small section of the soil instead of the complete soil structure, which is quite kind of a pity, because ideally from my first slides, it would actually be useful or interesting to actually find out a depth how plants grow, but this would be very difficult as you imagine, because we can't actually put an X-ray scanner down to the depths as well what we would want. In terms of future work, I think we would like to increase like the resolution to see finer features within the soil. This is common with synchrotron imaging, which we can actually do. And I think it would also be essential to have advanced imaging techniques which help us segment the plants from the roots better, because after taking the images, it's very difficult to piece out the roots from the soil. So to conclude, I would like to thank all the people who helped me. For instance, Manoj Menon and Stuart Carlson, who are my PhD supervisors, and some of the students I've worked with, undergraduate students I've worked with, who helped me with some of the images and processing, as well as I would also like to thank the Diamond Light Source and Neutron Immune Facility for actually helping us in terms of the image acquisition and everything else. Thank you for your attention.
Thank you. That's great, Tinashe. Really fascinating insights into actually what's happening below ground. You know, people are very focused on what's happening with the plant above ground, but you know, really important to try to look at those and the fantastic techniques to look at the 3D structures of those rooting systems under different soil conditions, you know, with different plant types as well. So excellent, really complementary to the stuff that Carsten is doing as well. So we've had lots of your questions coming in. Thanks very much. So I'm going to hand over now to Manoj, who's going to ask some of the questions on your behalf to the two presenters. Thanks, Manoj.
Yeah, thank you, Carsten and Tinashe, for the fantastic presentations. I have so far one question to Carsten about from Mandy Stoker, asking how can you differentiate between the carbon which is coming with the plant litter against the mycorrhizal derived carbon within an aggregate? Can you differentiate that? I mean, two different carbon pools, basically.
That's a good question. We haven't done so far. So I mean, like classically, we would like, I mean, you would, I guess, have two different replicates that where you can partition. I mean, like just going via the carbon source and getting this in via the plant, like for the synthetic carbon 13C, that's tricky. So it's like you would need different replicates, or maybe the source partitioning would be tricky, at least with nanosims. So,
yeah,
but this is an experimental setting. So, yeah.
Because you need to have a plant to have mycorrhiza as well, because maybe it's tricky to differentiate that way.
Exactly. So if you would really say, okay, how much comes direct, like you would say, like direct sorption or the direct input of purely plant origin OM versus what is like coming via the vector of the mycorrhiza, and to really partition that out. That might be more in the experimental setting, where you might have like different pots where you try to, I don't know, like get more longer. Length could be a thing that you kind of like, you cut diffusion and it is only the mycorrhiza that can reach the aggregate. But it's not in the imaging, it's not coming from the imaging. The imaging can't differentiate, at least not nanosims, that can't differentiate that. So that's in the experimental setting where, yeah.
Thank you so much. I don't have any questions yet. Nothing for Tinashe so far. Maybe if anybody has any question, please type it in that box, message box, so I can ask. We have probably another minute or so left.
That means everything was quite clear.
Yeah, you have been fantastic. Yeah.
Manoj, hi there. There is a question for Dr. Mawodza in the questions. Would you like me to read that out for you?
Yes, I didn't see that.
It's just been it's second from the bottom.
No.
Can you see that? Or would you like me to read it?
Yes, please. It's that one from Elizabeth Graham.
So it says, it's my understanding that neutron sources are not able to provide yet such high resolution as synchrotron CT. However, the possibility to distinguish different isotopes in CT is quite interesting. Have you contemplated using deuterated SOM or plants to trace them in the soil structure?
Yeah, that's to Tinashe.
Okay. Yes. So the resolution, of course, as you highlighted, for the neutron CT isn't as good as the X-rays, but there are people working on improving the resolution of the neutron CT. Because I actually don't work with organic matter in terms of the soil. I actually work more with organic matter within plants. I have used deuterated water, which is very distinguished between normal water and deuterated water, which actually helps us observe and monitor the water flow from the roots in the soil into the plants. So I actually have used it, although I didn't highlight it in the talk that I had.
Yeah, I think it's time almost. Sorry, there was another question which I couldn't take, unfortunately.
Okay, thanks. I'm sure there's probably lots of food for thought there, so additional questions probably come in after everyone's digested. But just want to round off really. So thanks very much on behalf of the British Society of Soil Science to our two fantastic speakers today, to Tinashe and to Carsten. some amazing research being done at the nanoscale and imaging. It's really fascinating. You know, we literally did zoom into soil today, so thanks very much for your contributions. It's really, really good. And also many thanks to Manoj as well, who organised the session on behalf of the regional group, the Northern Soil Network of BS-Cube. So thanks again to you, Manoj, for fielding those questions. And finally, thank you for everybody for attending. We've had a really good audience today. You'll find a quick feedback survey. Please fill that in as you leave the seminar. As I mentioned, this is being recorded, so check out the recording on our YouTube channel, as well as all the other Zoom into Soil webinars that have also taken place. We'd like to take this opportunity also to remind you that the Society is actually organising the next World Congress of Soil Science on behalf of the International Union of Soil Sciences, and the Congress theme is Soil Science, Crossing Boundaries, Changing Society. And we'll look at the focus on the link between soils and society, with sessions covering soil systems, processes, management, I guess lots of imaging now as well, and how we obviously interact with soils across the world. So do save your date, it's there on the screen. It's the 31st of July to the 5th of August in 2022. And as we say, in the meantime, do keep an eye on the British Society of Soil Science website for the forthcoming events. And we look forward to seeing you at the next Zoom into Soil, which is on Wednesday, the 9th of June. Thanks very much and goodbye.